Hello everyone, welcome back. So this is our last class for this particular course. Okay, so this is the week 12 of the NPTEL course, Experimental Biochemistry. This course is coordinated by Professor Dasgupta from the Department of Chemistry and Professor Dev from the Department of Bioscience and Biotechnology. And I am Beas Mukherjee, a research scholar from the Department of Bioscience and Biotechnology. And in today's class, we are going to talk about blotting techniques. Okay, so let's start with today's class. We are going to discuss northern blotting, southern blotting, western blotting, and all other associated blotting techniques in details. And then we are going to discuss a lot of previous year's questions. Okay, so let's start with today's class. Right. So what is blotting? Blotting is the process of transferring molecules. Okay, so right now we have a power shutdown, and that is why you can hear a lot of alarm going on. Okay, so sorry about this. Hopefully the power will be back very soon. Okay, so let's start with the class. So blotting is the process of transferring macromolecules such as proteins, nucleic acids, etc. from a gel onto a solid surface of an immobilized membrane to detect the molecules that have been transferred. So blotting techniques have a wide range of applications in molecular biology, genetics, biochemistry, biotechnology, and medicine. Okay, so what are the different steps? So it starts with protein extraction or DNA extraction, RNA extraction, whatever it is. And then there is SDS page. Okay, so this is the procedure for Western blotting. So there is SDS page and then finally there is result analysis. So after SDS page, the blotting and all the subsequent steps begin. Okay, until that it is the same. Okay, now blotting techniques such as Western blotting is extensively utilized in molecular biology to detect and quantify specific proteins in complex mixtures, aiding in the understanding of cellular processes and disease mechanisms. Okay. In forensic science, DNA blotting. Okay. So thankfully the power is back. Right. Yeah, perfect. Okay. Give me a second. I'll be back. Yeah, so let's continue. So Western blotting technique is extensively used in molecular biology to detect and quantify specific proteins in complex mixtures, aiding in the understanding of cellular processes and disease mechanisms. Okay, in forensic science, DNA blotting techniques such as southern blotting play a very crucial role in identifying genetic variations and establishing DNA profiles, supporting criminal investigations, paternity testing, etc. Okay, so this has a lot of applications, and that is why these techniques are very important for us. Now, what are the different applications? So, gene expression analysis. So, determining the expression of specific genes by detecting the mRNA transcript using northern blotting or quantitative RT-PCR, that is real-time PCR. Okay, and you have DNA detection and analysis. So detect and analyze proteins, their expression levels, post-translational modifications, interactions using Western blotting. DNA analysis, that is identify specific DNA sequences, analyze fragment size, study gene copy, number variations, and detect genetic mutations using southern blotting. Okay, so DNA analysis is done now. RNA analysis. So study mRNA processing, alternative splicing, RNA stability, small RNA species using northern blotting. Glycoprotein analysis. So if there are any kind of post-translational modifications in the proteins, this different kinds of blotting techniques can be useful in studying them. Okay, so detect and analyze glycosylated proteins and study protein glycosylation using Eastern blotting. Then there is protein-protein interactions which can be studied using far western blotting. DNA-protein interactions, these can be studied using southwestern blotting. Right? Then there is biomarker identification and drug development and forensic analysis. All of these things can be studied using the different blotting techniques that we have. Okay, now what are the different blotting techniques? So this can come as an important question. Okay, so let's try to understand these. So starting with northern blotting. So RNA is used to detect using northern blotting. Okay, now what is northwestern? Northwestern means protein RNA interaction can be detected. Far western is protein-protein interaction. Western blotting is proteins, detection of specific proteins. EMSA is also, no, but EMSA does not involve blotting, but then this is useful to study protein DNA interactions, okay, but then this does not involve any kind of blotting. Then we have southwestern blotting, where protein DNA interactions are studied, and then southern blotting, this was identified by, this method was developed by Southern. All of the names have been given because this particular one was Southern, okay. In all the others, you can see that the scientist's name is not Southern, okay. Right. So Southern blotting was developed by Southern and it is for DNA identification. Then to study the post-translational modifications, you have Eastern blotting. Middle Eastern blotting is for RNA DNA interaction. Far Eastern blotting is for lipid and post-translational modification detection. Okay. Now, what are the different types of blotting? So Southern blotting is used for detecting the target molecule DNA, and it detects specific DNA sequences, analyzes the DNA fragments, and then study gene copy number, identify genetic mutations, etc. Northern blotting, this is for RNA, detect specific RNA sequences, analyze gene expression levels, study mRNA processing, investigation of RNA splicing, etc. Then we have Western blotting, which is used for detection of the target biomolecule protein, detect specific proteins, analyze protein expression levels, identify post-translational modifications, study protein-protein interactions, detect and analyze glycosylated proteins, study protein glycosylation patterns. Okay. Now we have far western blotting. So for far western blotting, we have protein-protein interactions are detected using this. So labeled proteins are used as probes. So we will study about all of these techniques in details. Okay. Now we have southwestern blotting. Here we know that DNA protein interactions are studied. Okay. Right. So let's start with the different techniques. Okay. Now we have something very interesting, and that is called dot blot. So what is dot blot? DNA, RNA, proteins, antibodies, small molecules, all of these can be qualitatively analyzed using dot blot. Okay. So qualitative analysis of biomolecules screen for the presence or absence of specific sequences or molecules. That is done using dot blotting. So these small tiny round dots will be there, and if they are colored or if they are, if you see that they are dark, that means that particular thing is present. If not, then it is not present. So we'll study about this in details, right? Now we have slot blotting. What is that? This is for protein and DNA quantification. So quantitative analysis of DNA or protein samples screen for the presence of specific sequences or proteins without gel electrophoresis. Then you have reverse dot blot for DNA, RNA and protein using genotyping or identification of specific sequences by hybridizing labeled probes to immobilize target molecules. Okay, right. So let's start with the process of blotting. Oh, before that, we have the differences between northern blotting, southern blotting, and western blotting. Okay, so northern blotting is used for detecting RNA sequence, southern blotting for specific DNA sequence, and western blotting for specific protein sequences, specific proteins, right? So specific RNA sequence is detected. Okay, now what is the probe that is used? So cDNA or RNA probes are used. These can be radioactive or non-radioactive labels they can have, right? Now for southern blotting, single-stranded DNA probes or RNA can be used as a probe. Oligonucleotide probe is used. And for western blot, you know that antibodies against the specific protein of interest is used as a probe. Okay, so antigen epitopes. So the antibody is used against a particular antigen epitope, and that is the probe here. Okay. Now it is explained in details, so you can go through this, and the different applications are also explained here. Okay, so you can go through this. We will skip to the uh, steps involved in blotting. Okay, so it starts with a biological sample. Whatever is the biological sample, you take it, and then it is separated. So there is a mixture of different biological samples, let's say, and then you separate it using gel electrophoresis. So let's say if the sample is a protein, then you go for SDS page. Okay, everything else is the same. The normal protein preparation is done, sample preparation that you use for SDS page, that is done normally. Then after that, what happens is transfer from gel to the membrane. What membrane is this? For proteins, PVDF membrane is used. We will read about that. PVDF membrane very soon. For DNA and for RNA, NCM membrane is used. Okay. So nitrocellulose membrane, and this is PVDF membrane. Okay. Now transfer from gel to membrane by electric current or capillary action. So this is the blotting process. So transfer from gel to membrane is actually the blotting process. Now what happens after that? Okay, so a typical blotting experiment involves these 4 steps. Okay, so what are these 4 steps? Gel electrophoresis of the sample, transferring or blotting the electrophoresed sample from the gel to a membrane, treating the membrane with reagents or probe that bind to the target molecule and detect it on the membrane. This step is known as probing, right? Now the next thing is developing the membrane for visual detection, right? Here we will, this is the last step where we will see whether our protein of interest, DNA of interest, or RNA of interest is there or not, and how much of it is there. Okay, we can even quantify that. So one thing to note here is that the blotting technique does not involve electrophoresis. This is called dot blot. So the blotting technique, okay. Okay, hello and welcome. This is our last class, and we are talking about blotting in this class. Okay, so hopefully, yeah, good evening to you too. Okay, so let's carry on. So what is dot blot? Okay, so dot blot is very different from the other blotting techniques. How is that so? So dot blot does not involve any gel electrophoresis. Okay, so dot blot is a technique which is used in molecular biology and immunology to detect and analyze the presence or absence of specific biomolecules such as DNA, RNA, proteins, antibodies, or small molecules in a sample. It is a qualitative method and it does not involve gel electrophoresis, making it relatively quick and simple compared to the other blotting techniques like southern, northern, or western. For example, western blotting takes 2 days, okay, to develop, to complete as a process. Okay, now in a dot blot, the sample containing the biomolecule of interest is spotted. What is spotting? Dotted. Okay, small dots are made onto a solid support membrane, for example PVDF membrane or NCM membrane. The sample is applied in a small sample dots directly onto the membrane, and the membrane is then allowed to dry, okay, fixing the spotted biomolecules on its surface. Then what happens? Let's see what happens next. Okay, so after that what happens is, so we go back to the blotting technique, right? So we have transferred it onto the membrane. Now what? Now the membrane with the electrophoresed sample is onto it is transferred from the gel, right? So this is the actual blotting event that happens. Next, so the nucleic acid protein samples cannot be seen yet with naked eye, but then you can see it with a particular stain. What is this stain? This is called the Ponceau stain. Okay, so Ponceau. That is the spelling. So this is a red stain, and when you stain the membrane with this, you will see that all the proteins and everything else has been transferred. Okay. Now, next what do we do? So next so the blotting or the okay. So we are experiencing some network issues. Acha, so hopefully we are back again. Okay. So the blotting or the transfer step is done using one of two physiochemical phenomena. One is capillary action and the other one is electric current driven mobility. This is called electroblotting. Okay, so two approaches to the transfer step of a blotting experiment. So one is capillary action, the other one is electroblotting. So basically the transfer of all these samples or the bands of the gel onto the membrane. How are you going to make sure that it happens? So one way is through electric current. Okay, so that process is called electroblotting, and the other one is through capillary action. So electroblotting is faster. So what happens in electroblotting? Let's see. Okay, so we will look at both capillary blotting and electroblotting, right? So with capillary blotting, the experiment is arranged in such a way that the membrane is placed on top of the electrophorese gel. So dry paper towels that serve as blotting papers in daily life. So blotting papers are taken and they are cut into the shape and size of the gel. Okay, they are placed on top of the membrane along with the weight to aid in the blotting process, right? So along with the liquid buffer, technically called the transfer buffer. So this is the buffer into which all of this is drenched very well. Okay, the electrophorest macromolecule samples move from the gel to the membrane by capillary action. However, once the actual samples reach the membrane, they get stuck and cannot get beyond the membrane onto the paper towels. This is because the membrane is chemically modified to have special affinity for the macromolecules, so that once the samples reach the membrane, they get stuck onto the membrane. In the end of the capillary blotting step, the paper towels are soaked with the transfer buffer and the samples remain immobilized on the membrane surface. The transfer from the gel to the membrane is completed at this point. Okay. Now, in case of electroblotting, what happens? Now you know that the proteins are all negatively charged, right? Because you have run SDS page, so they are coated with SDS, so they are definitely negatively charged. Now, down below, there is the negative electrode and the positive electrode is on the top. So what happens when current is flowing, all the negatively charged molecules, they will be traveling towards the anode, which is the positively charged electrode here, right? So all the anions are going to the anode and the anode is on the top. So it will be traveling towards the top, and we again have network connection. Okay. So let's hold on for a minute. Hopefully it will be fine. Yeah, there was this problem with our network. We have a power shutdown right now. That is why there is no network. I'm somehow managing with my mobile network. So that is the situation. Right. So can you see? Yeah, your network is good. Here only we are facing issues. Okay. So let's carry on. Right. So we'll talk about electroblotting now. Okay. Hopefully things will be fine. Okay. So in electroblotting means blotting or transferring the sample from the gel to the membrane with the help of an electric current. Right. So this here is the working principle. So nucleic acids are negatively charged molecules. Also in SDS page, the net charge on the protein sample is negative due to presence of SDS. That is why the negatively charged molecules such as DNA, RNA or SDS coated proteins, they are exposed in an electric current, they will migrate towards the positive electrode or the anode. Right. So this phenomena is called electroblotting. Now, following electrophoresis, the macromolecule samples line the gel. Right. A membrane is placed on top of the gel. So this is your gel. Next, the membrane is placed, and before that also there are a lot of filter papers, and after this also there are a lot of filter papers. Right. So all the filter papers and everything is soaked in a transfer buffer, and then this transfer is happening. Now the gel membrane is sandwiched between two sponges, and this gel membrane sponge cassette is placed between two electrodes, so that the membrane is at the positive electrode side and the gel is towards the negative electrode side. The whole setup is immersed with plenty of transfer buffer for the electric current to flow properly. Now, due to the electric current, the macromolecule sample migrate or are transferred from the gel to the membrane. Right. Now, what happens after it has been transferred? Then what? Right. So let's look at that. Now we will add the primary antibody or the probe. Right. So before that, there is one more step called blocking. So what is blocking? So we will talk about that. Okay. Now the probe is being pipetted into the buffer solution. So it will be explained in details very soon when we come to Western blotting. So this is the basic blotting procedure. We'll look at this and then we'll go into the different types of blotting. Okay. So probe is being pipetted onto the buffer solution for binding to its target on the membrane. Electrophoresed nucleic acid or protein samples which cannot be seen yet with the naked eye, but their hybridization is happening. So once you add the probe or the primary antibody after blocking. So why is blocking important? So that this primary antibody does not go and bind to the non-specific regions, right? So that is why blocking is very important. Only your specific regions are exposed and there the probe will go and bind. Okay, so in case of RNA and DNA, this hybridization happens. In case of proteins, antigen antibody interactions happen. Okay, now next what happens? Let's see. So in nucleic acid blots, like northern blots and southern blots, the probe is typically single-stranded piece of nucleic acid that is complementary to your target fragment. This nucleic acid, single-stranded nucleic acid, binds to the nucleic acid fragment immobilized on the membrane due to nucleic acid complementarity. Just to put things in perspective, adenine A is complementary to thymine, guanine is complementary to what? Cytosine. Right? So thymine and uracil. Uracil for RNA, thymine for DNA, and GNC are complementary to each other. Right. Now complementary nucleotides bind to each other through hydrogen bonds. For protein blots, western blots and its derivative, for example, far western blot, northwestern, etc., etc. So the probe is primary antibody. You can see in the figure, right? So this is the target protein, and here is your primary antibody. Now this FAB region of the primary antibody will come and bind to the target protein, right? So paratope comes and bind to the target protein. The probe is the primary antibody and it detects a target polypeptide immobilized on the membrane. In most cases of the protein blots, the primary antibody probe is bound to the target is detected with a secondary antibody. Now the secondary antibody is often labeled, okay, and the secondary antibody comes and binds to the Fc region of the primary antibody, okay. So now the secondary antibody will be common for a lot of different primary antibodies, and the primary antibody is very specific to only your protein of interest, okay. Now the secondary antibody is either having some kind of chemiluminescent molecule attached to it or some radioactive uh, label, okay. So it is basically labeled, okay. Now we will look at that. Okay, so see what is happening. Treatment of the membrane with the probe that binds to the target molecule, probe being pipetted into the buffer, membrane embedded within the buffer solution. Final step is detection. So the sample image for the western blot, that is how it looks like, okay, and sample image for the northern blot, that is how northern blot looks like, okay. Acha. Now, what exactly happens? So, developing the detection signal. So, what happens in the detection step? So, this is the most important step, right? And for Western blot, after incubation with the primary antibody, you leave it overnight. The next day you come and then you wash it, remove the unbound antibody. Then you add the secondary antibody, let it incubate for 1 hour or so, and then once again you wash it so that all the unbound antibody is removed, and then you give the substrate. So the secondary antibody is conjugated with a particular enzyme. This enzyme reacts with its substrate to give a colorimetric product, and that color is basically quantified, okay. So we will look at that now. Developing the detection signal. So developing and detecting the signal for actual visualization of the target on the membrane is the final step in any blotting experiment. Simply put, the membrane is chemically treated so that the probe bound target. So one very important question is, what is the difference between a probe and a primer? Okay, so this very, like, a probe is also a oligonucleotide. Primer is also an oligonucleotide, but then what is the difference? You can tell me in the this what do you say, this chat window, okay. So what is the difference between the probe and the primer? Okay, you can tell me in the chat box, right? So we will carry on. So this ensures that the experimenter acha, you don't know, no problem. So primer, what is a primer? Primer is also an oligonucleotide that is used during PCR or polymerase chain reaction. So you have the DNA of interest or RNA of interest. Primer is also an RNA or DNA oligonucleotide. It will come and bind, and then what happens? Then you will add the polymerase and it will start amplifying, right? But what happens in case of a probe? Probe is also an oligonucleotide, which is once again single-stranded DNA or it is a single-stranded RNA. But probe often has a label to it. Okay, so it can either be radioactive label or chemiluminescent label or fluorescent label, but it will have a label for detection. But primer will never have any label to it. So that is the difference between a probe and a primer. Okay. So probe is used for some kind of detection method. For example, southern blotting, northern blotting, whatever it is. Okay. So that is something that you can remember. Okay. And not just blotting. Probes are used for a lot of different methods. For example, fluorescence in situ hybridization. There also hybridization is happening, right? So there also the probe will have some kind of fluorescence label. So there are different methods, microarray, this, that. Everywhere you will see that the probe is having some kind of label. Okay. Now we will talk about detection signals. So for visualizing the target on the membrane, the probe has any one of the following detection systems associated with it. One is colorimetric detection. So this is coming from color. Colorimetric color. Okay, so colored substrate will be, their colored product will be produced here. Okay, so now the probe is tagged with an enzyme. When the membrane is treated with a substrate for its enzyme, it is broken down into a colored precipitate which forms the colored readout for the target. Okay, fluorimetric detection. Here what happens, the probe is tagged with a fluorophore that emits fluorescence when excited by light wave of appropriate wavelength. The emitted fluorescence signal is the qualitative and quantitative readout of the target. Now we come to chemiluminescence. So in Western blot, this is also often used. Okay, so the probe is tagged with an enzyme, let's say HRP, which is horse radish peroxidase. Okay, when the membrane is treated with a substrate for this enzyme, it is broken down by a chemical reaction that emits light, and that is why it is called luminescence. Luminescence means giving out light, right? So this emitted light signal is qualitative and quantitative readout for the target. Radioactive detection. The probe is tagged with a radioactive atom. This is the most sensitive technique. Okay, so it is tagged with a radioactive atom. Technically, this is called radio labeling. So in nucleic acid blots, it is often a phosphorus 32 label and the radioactive isotope of phosphorus added in the sugar phosphate backbone of the nucleic acid probe. The probe, because of the radio label, gives out radioactive radiation. This radioactivity is captured on X-ray plate. It will come as a dark black band, right? And it is the readout for the target. Okay. Achha. Now we will look at this once again. So electrophorezed nucleic acid protein samples which cannot be seen with naked eye, onto it this your probe goes and sits, and the probe is, let's say, radio labeled. Then what happens? X-ray film is present. So the membrane with the radioactive probe is exposed to an X-ray film and band shows up on the X-ray film for the visual detection. So that small tiny black band will be there, and that is the presence of your DNA of interest. Okay, or RNA of interest. Right. So now we have two questions. Let's try to answer these two questions. Okay. Which of the following correctly matches blotting techniques with their detection targets and probes? So southern blotting, RNA and antibody. So detection target and probe. Okay, technique, detection target and probe. So for southern blotting, which of these is correct? Right, try answering. For the next question, a researcher wants to detect a specific mRNA using a blotting technique with high specificity. So which one of these would be most appropriate? So Western blot plus primary antibody, Northern blot plus complementary nucleic acid probe, Southern blot and antibody against DNA, and immunofluorescence with fluorescent DNA probe. So for mRNA of a particular sequence, which one would be most appropriate? Okay. Chalo, try answering. This is very straightforward question. C. Yes, it is C for the first question. Good. What about the second question? Acha. So, for RNA, it is always northern blot. Right? How will you remember? DNA, southern blot. Protein, western blot. RNA is always northern blot. And what will be the probe then? Of course, it has to be complementary sequence, either DNA or RNA. Right? So, complementary nucleic acid probe. That much is clear, right? And it is always northern blot for RNA. So I'll go back to that slide because when I started, you were not there. So this slide will help you to remember. Okay, this is the slide. This is called the blotting compass, and here you can see that northern is for RNA, southern is for DNA, because southern, a scientist with the surname Southern actually developed this technique. Okay, developed blotting technique for DNA. So to, like, for humor, the other names were then given. For example, northern for RNA, western for proteins, Far western for protein-protein interactions. Northwestern, that means protein-RNA interaction. Okay, southwestern is DNA-protein interaction. And EMSA is given here, but it is not a blotting technique. And then we have eastern blot. So what is eastern blot? This is for lipids. Okay, so eastern is for post-translational modification. Far eastern is for lipid and post-translational modification. Middle eastern is for DNA RNA interaction. And we have eastern western for lipid and protein. Clear? So this is the whole story of blotting. Right. So now we will go back to the question. Okay. So the right answer is it not interesting? Do you not find this interesting? Okay. So the answer is here. So northern blot detects RNA using complementary labeled nucleic acid probes. Okay. Right. Now, southern blotting procedure. Okay. So what is there in the southern blotting procedure? Let's see. I will zoom in so that you can view nicely. Okay. So initially what is there? A gene of interest is there, right? So DNA containing the gene of interest is extracted from the human cells and cut into fragments by restriction enzyme. So only our DNA of interest is excised out, right? Then what happens? The fragments are separated according to their size. So basically you run an agarose gel by gel electrophoresis, and each band consists of many copies of particular DNA fragment. The bands are invisible to the naked eye, but they can be made visible using staining. Now, next what is that? There is this buffer solution, a lot of filter papers. On that you place the gel, then you place the membrane. Once again a lot of filter papers, and the buffer solution is there, and you place the electrodes, and then the current is passing, and your sample is moving onto the membrane, to the NCM nitrocellulose membrane. It is sticking to the membrane at the exact same positions as it was on the gel, right? So it is just a replica. So the DNA bands are transferred onto nitrocellulose membrane by blotting. The solution passes to the gel and filtered to the paper towels. Now this produces a nitrocellulose filter with DNA fragments positioned exactly as on the gel. Then what happens? The filter is exposed to a radioactively labeled probe. So see, probe is labeled. Always remember, probe is a oligonucleotide that is labeled, right? So probe for a specific gene, and the probe will hybridize, right? So it will hybridize, that means it will bind. Base pairing will happen, hydrogen bonds will form with the gene of interest, it will hybridize and with a short sequence present on the gene. And then the filter is exposed to X-ray film, and the fragment containing the gene of interest is identified by the band developed on the film. So a gene of interest will be visible because it is radioactively labeled, right? So so when you expose it to X-ray film, it will give this black band. Now, the next one is Western blotting. So let's see what happens in Western blotting. Okay. So in Western blotting, you run an SDS page. Until then, all the sample preparation, everything is exactly as you do for SDS page. So you run the SDS page, you take the gel, you don't stain it. You take it and then you go for the blotting. So what is that? Filter paper, gel, membrane, filter paper. And electrodes are there, so current is passing and the transfer happens. So basically blotting onto the PVDF membrane is happening. Then what do? Then you block the membrane. So using BSA. What is BSA? This is bovine serum albumin, right? So bovine serum albumin or milk casing. So we also use SM powder. So what is SM powder? That is also milk powder, normal milk powder that you get, right? So using BSA, using SM powder, whatever, you block the membrane. Okay. Now, next what is that? Primary antibody. So you add the primary antibody. So primary antibody, remember, it is complementary to our protein of interest. It is going to bind to the epitope of the protein of interest. Correct? Now, then you wash and remove all the unbound antibody. Then you then you let it incubate for overnight. Then you add the secondary antibody. And then once again you let it incubate for around 1 to 2 hours. Then you wash again. So incubate the membrane with HRP conjugated secondary antibody. So what is HRP? It is an enzyme. Horseradish peroxidase enzyme. So this secondary antibody binds to the Fc region of the primary antibody. So that means if the primary antibody is produced in, let's say, mouse, then the secondary antibody will be complementary to all the primary antibodies that are produced in mouse. Correct? It is complementary to the Fc region. Okay. Now, the next one is you will have to develop the blot. So then again you wash, all the unbound secondary antibody is removed. Finally you wash. Okay. So incubate the membrane with chemiluminescent substrate. So you add the substrate. Now this enzyme is there. Enzyme is reacting with the substrate. A product is getting formed. This light is released and we will look at the reaction now. Okay. And you see using autoradiography, you see that there is this band. So using our ChemiDoc machine, you detect the presence of this black band. And that will tell you that our protein of interest is present. How much of it is present, that can also be quantified. Okay. Now exactly what happens in this reaction. So let us look at that. So in this reaction, let's see. In chemiluminescent Western blotting, the enzyme catalysis reaction involves the oxidation of luminol. So luminol is the substrate. So oxidation of luminol by the enzyme horseradish peroxidase. It is taken from a plant, okay, and in the presence of hydrogen peroxidase, hydrogen peroxide, okay. So here's the reaction. So there is luminol, there is hydrogen peroxide, and that forms this 3-amino phthalate and this 4-hydroxy 3-amino phthalate and light. So in this reaction, luminol is a chemiluminescent substrate. It is a derivative of the phenyl hydrazine and it emits light when oxidized. H2O2 is serving as the oxidizing agent. Horseradish peroxidase is the enzyme that catalyzes the reaction. HRP is commonly used in Western blotting due to its high specificity and efficiency in catalyzing the peroxidation reaction. The reaction produces an excited state of 3-amino phthalate along with 4-hydroxy 3-amino phthalate and light. So the excited state, so this star is given, so that means this 3-amino phthalate is in its excited state, right, not in the ground state. So in the excited state, it is subsequently returning to its ground state. So that means the energy is getting released and the electron is coming back to the ground state, emitting light in the process. So we know that in fluorescence and in phosphorescence, we know that the electron returns from excited to ground state by giving out light, right? So this energy, this emitted light is then captured and visualized using the X-ray film or chemiluminescent imaging system. This is the ChemiDoc system that we use, allowing for detection and quantification of the target protein in the Western blot. Okay, so the light is basically detected and quantified. Okay, now we have a lot of questions, so let's start answering them. So for the first one, in southern blotting experiment, a researcher observes multiple bands even though the genomic DNA was digested using restriction enzyme and cut only once within the target gene. Which of the following is most likely explanation? So there can be partial digestion of the genomic DNA, presence of introns in the gene, cross-hybridization of the probe with the homologous sequence, and degradation of the RNA during transfer. Genomic DNA was digested. So genomic DNA means that there is no RNA, right? It's DNA only. And they are saying that it was cut only once within the target gene. but then multiple bands are there. So what is going on? A, partial digestion. Okay. For the next one, so which of the following steps is unique to Western blotting and not required in Northern blotting? Transfer to the membrane, blocking of the membrane, hybridization with labeled probe, and gel electrophoresis under denaturing condition. So which is not required in the Northern blotting? Transfer to the membrane would not be the right answer because blotting means transfer to the membrane, right? So it would be there in all of the blotting steps. Correct. Gel electrophoresis, hybridization with labeled probe, blocking of the membrane. Which one do you think is the right answer? See, let's see. Okay. So for the first one, so most likely explanation is for cross hybridization of the probe with homologous sequences. So it is possible that there are a lot of homologous sequences in the genomic DNA, right? And so it is possible that it is binding to all of these homologous sequences, and that is why possibly your so many bands are there, because the probe, remember, it is very specific to the gene of interest, right? And if there are more than one copy of the gene of interest and they are of different sizes maybe, then it will give multiple bands because it will go and bind to all of them, right? The next one is the blocking step. So remember, for proteins this blocking step is necessary, and for northern blotting and for western blotting this is not necessary as such. Okay. Now, northern and southern blotting. Okay, it is necessary for western. Now, see, a researcher performs western blotting and observes a band at a higher molecular weight than expected. So, what is the most plausible reason? So, western blotting means it's a protein. Okay, so there is a band at a higher position. So, that means what is going on? Post-translational modification, for example, glycosylation, DNA methylation, incomplete restriction digestion, alternative splicing of mRNA. Remember, this is Western blotting. B. Okay, great. Now try the next one. In Western blotting, a researcher switches from chemiluminescence to fluorescence detection. Which of the following changes is most likely? An increased signal amplification due to enzyme turnover, ability to perform multiplex detection of protein, required of radioactive probes, loss of specificity due to antibody cross-reactivity. Okay. So we will look at the right answers. Acha. Now, the first one, it is correct. So post-translational modification, for example, glycosylation, it will increase the molecular weight of the protein. So it is going to run higher than its expected place, right? So that is correct. What about the second one? If there are, let's say, one protein is, let's say one of the antibodies, it is bound to RFP, red fluorescent protein. The other one has no, let's say that there are more than one fluorescent proteins, right? And it is actually, yeah, so basically it is, so if there is fluorescence detection, so one of the conjugates, it gives you red fluorescence, the other one gives you green fluorescence, the other one gives you, let's say, yellow fluorescence, right? So YFP, all of these different proteins are there, and then what will happen, you will be able to identify whether all these three different proteins of interest are there in the same sample or not. So three different primary antibodies will be used, and different secondary antibodies will be used, and you can go for multiplex detection of proteins. So that is called multiplex detection of proteins. Okay, so at the same time you can detect more than one proteins if you have more than one set of primary and secondary antibodies. next one. Why is chemiluminescence generally more sensitive than colorimetric detection in blotting? So why is this more sensitive? Okay. So it uses radioactive isotopes. Enzymes generate multiple photons per substrate turnover. It eliminates the need for secondary antibodies. It directly detects DNA without probes. So when we are talking about chemiluminescence and colorimetric detection, it is definitely not involved with DNA, correct? So this gets cancelled. Now you choose amongst these three. Again, radioactive isotope, if it is chemiluminescence, there is no radioactivity, right? Then it would have been radioactive isotope. So why is this chemiluminescent one more? Yeah, so enzymes generate multiple photons per substrate turnover. Okay. What about the next question? In a Western blot, improper blocking leads to high background. Which of the following is the best mechanistic explanation? Okay, so if your blocking is not for sufficient amount of time, you see that the entire blot is like blackish, right? So why is that? Increased degradation of the target protein, non-specific binding of the antibody to the membrane site, loss of antigen during transfer, incomplete electrophoretic separation. Acha. So increased degradation of the target protein, non-specific binding of the antibody to the membrane site. So blocking is done before addition of the primary antibody, right? So before addition of the primary antibody, you want to make sure that all the non-specific sites are blocked by this BSA, right? And only your protein of interest is uh, present and it is exposed to the primary antibody so that the primary antibody comes and binds to it. If the blocking is not done properly, primary antibody will possibly come and bind anywhere, although it is very, very specific, but it is a protein. So there is a possibility that there will be other non-specific sites where this protein might come and bind. Then what will happen? Your secondary antibody is designed in such a way that it will definitely come and bind to the primary antibody. Now, if the primary antibody is bound to places other than the protein of interest, secondary antibody will also go and sit, right? Then when you image it, when you add the substrate and you image it, you will see that instead of single band, there will be a lot of black thing all over, because all those regions are giving signal now, right? So there will be a lot of non-specific binding of the antibody to the membrane site. Loss of antigen during transfer? No. Why? Because the antigen is immobilized on the membrane. It is not going anywhere. Incomplete electrophoretic separation, this is not valid. Why? Because electrophoretic separation has already happened during SDS page. So after that, it is not going to change, right? Increased degradation of the target protein. Degradation of the target protein is not going to happen if your blocking is improper. If the protein is degrading, then you will not see signal, or you will see bad signal. That is a different story. Okay, but due to blocking, this is not going to happen. Okay, so the right answers are B and B again for the two questions. Okay, right, now we have more questions. Let's try to finish them up. So what is the first step of the Western blot? To run agarose gel, to run SDS page, to run proteins in a membrane, to transfer proteins in membrane, to add primary antibody to the sample. And Western blotting is performed to detect specific DNA, specific RNA, specific protein, specific protein DNA complex. Which one? Very straightforward questions. B, excuse me, B and C. Okay, good. Yes. So the right answer is definitely B and C. Okay. Now, Western blotting technique is known as Southern blotting, hybridization, immunoprecipitation, and immunoblotting. Western blotting is known as what? So in which subject do you study about antigens and antibodies? Which which part of biology do you study about these? The proteins in a sample are separated using. So proteins are separated using SDS page, Western blot, protein purification and immunoblot. So antigen and antibody you study about where? Try answering. Acha. So basically in immunology, you read about antigens and antibodies, right? So that was a clue. Anyway, so Western blot uses primary antibody, secondary antibody specific to your antigen, right? Epitope of the antigen. That is why it is called immunoblotting. Okay. The proteins in a sample are separated using SDS page. So we will look at the right answers. So see, immunoblotting. So Western blotting, remember it is also called immunoblotting. Okay. Why immunoblotting? Because definite antigens are detected using definite antibodies. That is why it is immunoblotting. Okay. Proteins in a sample are separated using SDS page. This you know. Okay. Now, Achha, I am not familiar with this kind of topics. Okay. Alright. Achha. So for a DNA fingerprinting, which of the following technique is used? Okay, so for DNA fingerprinting, what is DNA fingerprinting? So DNA fingerprinting means that specific regions in the DNA are identified using very specific probes, and that helps us to find out. So DNA fingerprinting is very useful in detection of criminals. For example, some murder scene has happened and you know that all these forensics people, they go and they collect items, right? And then they go for DNA fingerprinting to identify who this who has touched all these things, right? So basically whether it is belonging to a particular person or not. Then there is paternity testing, so whether a particular person is the father of another person or not. So paternity testing, that is done, right? So DNA fingerprinting, then which technique do you think it uses? Southern blotting, northern blotting, western blotting and dot blotting. For the next question, in blotting techniques, a probe is a specific protein to detect DNA. The answer, I'm showing you the answer. Okay, so there was no question. Yeah, the answers are visible, I realize. Anyway, so we will just look at this and go to the next question. So a short piece of labeled single-stranded DNA or RNA which is complementary to a specific DNA or RNA is called a probe. Okay, in blotting technique. And DNA fingerprinting is for southern blotting. Okay. Now, which of the techniques? So, next question, you can try to answer this one. So, which of the technique is suitable for detecting a specific mRNA expression in a cell? So, specific mRNA expression. Now, try to answer. The next question is, the non-specific sites on the PBDF membrane can be blocked by Twin 20, non-fat, dry milk, sodium dodecyl sulfate or SDS. Acha. So 2 is B. Okay. What about the first one? Acha, yeah, good. So it is also B. So for both, the right answer is B. So this non-fat dry milk is also called SM powder. Okay. Why the probe is labeled? Why is probe labeled? To improve binding capacity, to improve location identification, to improve visibility, to improve stability. What is the major force behind protein or nucleic acids to go out from the gel onto the membrane? Electric current, pH gradient, voltage and temperature. Which one do you think? B. B is to improve the location identification. Probes, location. Okay, so we will look at that. Now, what is the major force behind proteins and nucleic acids to get blotted onto the membrane? Electric current. Acha. So, we'll look at the right answer. Okay. So, basically, yes, to identify the location, for location identification. So, it will go and sit, right, somewhere. And where it is sitting, that we want to know. So, that is why location identification is the right answer. For the second one, this negatively charged molecules, they are all traveling with the current and they are going to the positively charged electrode. In between, they are meeting the membrane. Membrane is chemically modified in such a way that it will stop all these negatively charged molecules, be it DNA, RNA or protein, and that is why this blotting happens, right? So blotting happens because of electric charge and because of voltage difference. Only when there is voltage difference, electricity will flow, and only when electricity flows, then what happens? All these negatively charged anions will move towards the anode, and in between they will meet the gel where they'll go and stick. That is how we get the blot. Okay, we have a few more questions and then we will be done with the whole course. Okay, let's try answering. Why labeled antibodies are used to detect the presence of particular DNA molecule in northern blotting, detect the presence of particular RNA molecule in southern blotting, presence of particular protein molecule in western blotting, and detect the presence of particular DNA molecule in southern blotting. What about the next one? 2B. So for both it is B. That is what you're saying, right? Acha, 2B. So which of the following is used for detection of protein in western blotting? So antibody is used. Correct. Yes. What about the first one? Try answering the first question. 2 is B. That is correct. C. Yes. Right. So it is C and B for both the questions, right? Okay, great. Now we have, I think, a few more questions left. Okay. So question on blotting once again. Which of the following membrane is best to use if you want to strip an already used membrane for western blotting? So that means you want to remove all the immobilized protein and everything from that membrane. That is called stripping, right? So then what membrane should you use? There is nylon membrane, there is nitrocellulose membrane, there is PVDF membrane B. Okay. Why ghost bands appear in blots? So what is this ghost band? So ghost band means there is some kind of non-specific band. Okay. Too much protein has been loaded. Blocking was not done. Incubation with primary antibody was too long. Too much substrate for the secondary antibody was added. So that means, let's say a band was supposed to be here, but then we are seeing one here, one here, one here. Okay, so nearby it there is a lot of bands. These are the ghost bands. Why do you think they can be there? One is B. Okay. What about the next one? There can be more than one right answers. Okay. So we'll look at the right answer. Okay. Action. So too much protein, if that has been loaded, then it can appear as a ghost band. Okay. Action. And if the blocking is not done and if incubation with the primary antibody is too long, then you will see a lot of non-specific bands as well. Okay. Right. Now what is this difference between PVDF membrane and NCM membrane? we will look at this now. Okay. Now, one very interesting thing about NCM membrane and basically nylon membrane is that, see, if you are using nylon membrane, then this DNA of interest and RNA of interest will go and stick to the nylon membrane. But for nylon membrane, you will have to heat it for fixation, right? For proteins, you don't need to heat them for fixation to the membrane. But for DNA and RNA, you will have to heat it for a temperature of up to 80 degrees centigrade for some amount of time to make sure that they are stuck. So that is a very important thing that you should remember. Now see, what is PVDF? Polyvinyl diene polyvinylidene difluoride. So PVDF. The PVDF membranes have high protein binding capacity and are suitable for a wide range of protein, including those with low molecular weight. They have high mechanical strength and chemical resistance, making them suitable for harsher experimental conditions such as stripping, reprobing membranes multiple times. PVDF membranes are compatible with various detection methods including chemiluminescence and fluorescence. What about nitrocellulose membranes? Nitrocellulose membranes also have high protein binding capacity and are generally more cost effective than PVDF membranes. Excuse me. They are suitable for proteins of moderate to higher molecular weight and are commonly used for protein blotting applications. NCMs are more prone to degradation under harsh conditions and they cannot be stripped as easily done with PVDF membranes. In summary, both PVDF and NCM membranes are widely used for protein blotting. PVDF membranes offer more advantage in terms of versatility, durability, compatibility and various detection methods, whereas nitrocellulose membranes are more economical but they are less robust and under harsh experimental conditions they will get degraded. The choice between these two depends upon the type of protein studied. Okay, so remember that. So PVDF is for smaller molecular weight proteins and NCM is for larger molecular weight proteins. Now this is our last question. Last two questions for this full course. Try answering. If you forget washing the membrane after incubation with primary and secondary antibody, what will happen? Background noise will be very high, whole blot will be dark, no specific band will be observed, no change in the blot will occur. Okay. And for the next one we have, in far western blotting, a researcher aims to identify proteins that interact with a known transcription factor. Far western means protein-protein interaction, right? So which of the following strategies is most appropriate? So use of a labeled probe complementary to the transcription factor gene, use of a primary antibody against the transcription factor followed by enzyme linked secondary antibody, incubation of the membrane with labeled recombinant transcription factor protein. Okay, so one is B and C and two is you don't know. Okay, great. Let's look at the right answer. Yeah, so yes, the whole blot will be black if you forget to wash, because primary antibody will go and bind wherever it wants. Secondary antibody will once again go and bind to all the primary antibodies and you will get a black beautiful blot. Okay. Now, what about the next one? So, incubation of the membrane with labeled recombinant transcription factor protein. So, in the far western blotting, the probe is not an antibody but a labeled protein. Okay. The goal is to detect protein-protein interaction. So let's say this transcription factor interacts with another protein. So this another protein will be there and this another protein will be labeled and it will come and bind to this transcription factor. So we are identifying protein-protein interaction. So the immobilized protein on the membrane are probed with a known interacting protein. Okay, often tagged or labeled. So that is the full story of far western blotting. And that is all about this course, Experimental Biochemistry. So all the references are given. All the best for your exams. Okay, do your very best. If there are any questions, you can mail me, you can ask me. I'm always here for answering. Okay, good luck. So if you don't have any more questions, we can end the class. Yes, this is our last class. So yes, thank you so much for attending the classes. Thank you so much. I hope you have been able to learn something out of these classes. Thank you so much. Yes. Bye bye. Yes, bye bye. Have a great life ahead. So I'm ending the call for everyone.